Welcome to solverfy.
So in this video, we're going to be going over the Anabaena and heterocyst development flowchart again. And basically what this is saying is if there is low or absent fixed nitrogen in the environment, then that is going to cause increased free calcium and 2-oxyglutarate concentrations. And then with these two increase, that would stimulate expression of genes that produce these two transcription factors, NTCA and HETR. And both NTCA and HETR are going to promote heterocyst development, which you can see right here. And then in addition, HETR is also going to promote production of PATs. And PATs is going to inhibit heterocyst development in adjacent cells. So you will start developing heterocysts, but it's not going to be every cell because PATs is going to inhibit adjacent cells from developing into heterocysts. So now we're going to be looking at this chart and we're just going to be altering a couple things to see what happens to the end product. So first what we're going to look at is if you decrease the free calcium, so this is looking at right here, if you decrease the free calcium, is that going to prevent or cause heterocyst development? Well, we know if we have increased free calcium, that's going to cause heterocyst development. So if you decrease this free calcium, then that should prevent heterocyst development because you wouldn't have these two transcription factors being produced, so you wouldn't have this heterocyst development occurring. So it should be prevent. And next we're going to look at, in the presence of fixed nitrogen, is NTCA going to be expressed or not expressed? So in this example, it's looking at low or absent fixed nitrogen. And the question is asking about if there is a presence of fixed nitrogen. So if there is a presence, then there isn't going to be increased free calcium. So let's just say it's going to be decreased free calcium. And if there is decreased free calcium, then it's not going to stimulate the expression of NCTA or HETR. So in the presence of fixed nitrogen, NTCA should not be expressed. Okay, our next example is again going back to low fixed nitrogen in the environment. Should that increase or decrease PATs concentration? So here we have, and I'll zoom out a little bit so you can see, here we have low fixed nitrogen environment. That's going to cause the increase calcium and 2-oxyglutarate, which is going to cause the production of these two transcription factors. And then if you have HETR being produced, then that's going to promote the production of PATs. So in a low fixed nitrogen environment, you should have increased PATs. And then for our last example, this is going to ask if you have a mutation making NTCA non-functional, would this allow adjacent cells to develop into heterocysts? Well, we know NTCA is going to cause heterocyst development. However, PATs is what's going to inhibit heterocyst development in adjacent cells. So if HETR was non-functional, then that would allow heterocyst development in adjacent cells. But it's not asking about HETR, it's asking about NTCA, which doesn't affect the allowing or inhibiting of heterocyst development in adjacent cells. So this would actually be false.